So we'll start today with a shameless plug for one of the other courses that I'm teaching next term. Virology is one of them. The other one is mutant viruses from hell, also known as recombinant DNA laboratory techniques. So this is a lab which we'll be teaching down at the CLSB. Basically, what it is research projects from my lab, because we do work on these mutant viruses from hell. And basically the way that the course works is that first day you get a couple of mutants. We have no idea what those mutants are. In fact, they may be very useful to our research at some point. And then you tell us what those mutants are and what they do. And so I just told you that in about 20 seconds. It takes about 10 weeks to do all that. But you learn a lot of laboratory techniques, and this is a real research project. We'll also be talking a lot about the scientific literature. It goes behind all of these things. There'll be presentations as well of your results, and then talking a little bit about the scientific literature. And I just recently found out it's the cure for the Common Lab, because it's a course-based undergraduate research experience. Here's the CRN. This is posted on D2L, so you can take a look at it. Any questions, just send me an email. So with that, we'll move on to everybody's favorite part of class. Yes? Our clicker questions from last time. During homologous recombination, Rec-A first binds to single-stranded DNA with a 5' end, double-stranded DNA, single-stranded DNA with a 3' end, RAD51, or pCNA. I know there are more than 66 people here. Ten, 5. Doing my best. Okay, so let's take a look at what you thought. It's on the wrong screen here. Trying to reset this such a way that 5' end, 3' end. I'm not in the way of everybody when I'm in front of the screen here. So during homologous recombination, Rec-A, nobody liked RAD51. Why not? Because it's the Rec-A from yeast, exactly. Nobody liked pCNA. Why did they not like pCNA? Okay, that's also a very good reason. What if it were beta? A sliding clamp. It has very little to do with this process. So we're down to single-stranded and double-stranded DNA. What happens after you have a double-stranded break if it's going to get recombined through homologous recombination? What happens? First thing that happens after you have the double-stranded break. You have exonuclease that chews in at the ends. What end does it chew in on? It chews in the 5' end and leaves you the 3' end. And that's what binds to Rec-A. So, yes. Our second clicker question. Gene conversion happens because of incorrect proofreading, base excision repair, nucleotide excision repair, non-homologous end joining, or mismatch repair. Again, feel free to discuss. 10, 5. I'm sure people didn't leave since the last clicker question. Or maybe they did. Distinctly possible. So what is gene conversion? Something that Stedman made up for this class, so we had a clicker question. So you have allele switching that takes place. So either the maternal copy is passed along more than it otherwise would be, or the paternal copy is passed along more. And that's because you have what? You have homologous recombination, right? So that's not one of the possible answers here. Because the homologous recombination, they're homologs. They're not identical to each other. So what are you going to have on opposite strands relative to each other? You'll have mismatches, which are not the appropriate base pairs. So how do you repair them? Mismatch repair, which, again, most of you chose, which is great. I did a great job of explaining it, or the question was really easy. You can take your pick.
It's not easy.
Okay, I just wanted to review really quickly what we talked about last time and then move on and talk about transcription. Last time we talked about really 3 kinds of jumping genes. What we didn't talk about and what's at the very end of the lecture is not going to be covered probably on the next exam unless I find a way to shoehorn it into some of our other lectures. So stuff that is not covered in lecture will not be on exams. So don't worry too much about lambda recombination and Cree recombination. Even though it's totally cool and fascinating, I'm happy to talk about it offline. So what we did talk about, however, are the DNA transposons, which have the inverted repeats at either ends, have a transposase, and they pick up that DNA and move it and plop it in somewhere else. This happens a lot in bacteria. There are a few in eukaryotic cells, but the vast majority are going to be in bacteria. And the real problem medically, often they also have antibiotic resistance genes as well as the transposase gene that's part of them. Retroviruses, some of the coolest viruses ever. Zika is not a retrovirus, just in case you were wondering. I just posted a link at how Zika virus did not recombine with mosquitoes, has absolutely nothing to do with it. That's been floating around the internet. In fact, there's a great article from the Guardian newspaper which said, you know, the next problem is the viral expansion of Zika conspiracy theories. Quite a nice read. So, but these retroviruses, what they do is they then, through the activity of the reverse transcriptase, will take an RNA copy, make it into double-stranded DNA, and put that into the genome. Once that's put into the genome, that gets transcribed by all the stuff we'll talk about later today. And that RNA encodes a reverse transcriptase, which can, you know, take that RNA, which has been transcribed, make it into DNA, put it back into the genome. And so there are many, many copies. There's about 8 to 10% of our genome are these retroviral-like integrated sequences in our genome. And so that's way more than the approximately 1.5% of our unique protein coding genes. Retrotransposons we understand a lot less about, but basically what they are genes that code reverse transcriptases, but instead of having all of this extra viral-like replication process, they basically make a copy of their own gene and pop it into the genome, but it's a messenger RNA. It's got this whole long poly A tail at the end. And so poly A tails, if you're making double-stranded DNA, are going to have AT base pairs at the end. So that's the easiest way to tell. That is by far and away the majority of the repeated sequences that we have in our genome, are these non-retroviral retrotransposons. So questions about that before we move on and talk more about some of the rest of it. So just the outline of what we did talk about. Again, we've just talked about site-specific recombination, homologous recombination. Why do we do homologous recombination? What is the reason for homologous recombination? So stopping mutation, but that's sort of an extra step. That's your meiotic recombination. Why? What does homologous recombination do? It fixes one particular kind of DNA damage, which is double-stranded breaks. Those double-stranded breaks get made specifically in meiosis to allow for meiotic recombination to take place. But probably someone asked me after lecture last time about this. It's highly likely that meiotic recombination evolved from a double-stranded break repair mechanism that then was taken over to allow this meiotic recombination process to take place. Mechanisms, again, what happens, mentions briefly in the clicker question, double-stranded breaks, cleave in 5' ends. You've got an overhanging 3' OH that associates with some kind of single-stranded DNA binding protein, REC-A, RAD51, that then undergoes strand invasion, finds a double strand, pokes itself in there. Now it's got a template. Now it's got a 3' OH that can be extended. That then generates a holiday junction. We've got crossed over strands relative to each other, more like a kind of an X structure. That then gets chopped and put back together, which will either give you crossovers or patching mechanisms. Questions, comments, worries? Is it going to be on the exam? Yes. Oh. So now move into some of my favorite subject matter for the term, because I spent 6 years of my life doing this, is transcription. So transcription is all about this second step here. So we talked about now DNA replication, repair, recombination. Now we're talking about transcription. So it's taking the DNA and making an RNA copy from it. Talk a little bit about RNA. Most of the time we'll talk about the enzyme which does this, which is the DNA-dependent RNA polymerase. Now we'll talk a little bit about how bacteria start, how eukaryotes start, and it turns out that the start here is absolutely critical for what genes get turned on or turned off, because that's going to be what's making your RNA. And if you've got time at the end, we'll talk about my now current favorite organisms, the archaea. So if you look at everyone's favorite chromosome, this is the X chromosome, human X chromosome up here. We talked a little bit about this already. This is, in fact, unlike the chromosome 22 that we talked about last time, this is one of the largest human chromosomes, 155 million base pairs, about 5% of the whole genome. We're now zoomed into a really tiny part of this chromosome. All the vertical lines here, the gray ones, stand for exons. And again, just like we've seen before, exons are really in the minority. Exons are what's in fact coding for protein. The other thing you can see on here is where we have changes relative to Neanderthals. And the basic take-home message here is very, very few. All these triangles here. And probably most important, as far as this slide is concerned, is that if you look here in terms of your conservation or homology, oops, that's interesting, it's really quite variable. So everything here, the blue lines at the bottom, these represent, if you've got a line that goes all the way to the top, that means in 100 different vertebrate genomes, that sequence is exactly the same. And that means it's probably likely to be really, really important for function. It's been conserved for very long periods of time. When you can hopefully see here is that these blue lines are clearly a lot higher where you have exons, but it's also variable. So it depends on what you have here. How do you get all of these? Again, each of these represents a set of exons, but the gene name is up here. These are clearly things you have to put together. We'll talk a lot more about splicing later on. One of the big issues, if you just look at a genome like this, is how do you predict that this is actually making a protein? And the answer is it's really hard to do this. And that's one of the big problems that people have had in terms of interpreting the genome sequence. Did anybody go to Mark Tashney's talk last night over at Reed? Yeah, it was interesting, wasn't it? It was pretty hilarious. But basically, he said that genome sequencing is worthless. Is a good paraphrase? Yeah. I think part of the reason for that is that it's really hard to, by just looking at sequence, know what's going on there. If you don't have the sequence, there's nothing you can really do about it. So he kind of ignored that part. But a lot of it has to do with being very hard to predict what the genes are. The other issue, and that's what we have down here with these little diamonds, is there's also lots and lots of non-coding RNAs that turn out to be really, really important for function. Here, if anything, in fact, the number of non-coding RNAs is far too small. This one only has, let's see, about 6 on it. There are probably way, way more than that, and that's, again, one of the latest things we found in looking at the genomes that are totally worthless, according to Mark Tashney, is that there are as many non-coding RNA genes as there are coding RNA genes. And so that's a really big issue with looking into that. Now, how do you find out about this? One of the main ways is actually just to look and see what is, in fact, being made into RNA. And a lot of what we do now know about these small RNAs is that there's a heck of a lot of them, in fact, way, way more than we ever thought was true. Yeah?
By non-coding RNA, you mean it is not going to be translated into a protein. It is going to have some second, what we consider secondary function.
Yeah, so when I say non-coding RNA, it really should be non-protein coding RNA. And that's the terminology which people use for that. Yeah, code, if you want to use code, it could be all kinds of different things, but non-protein coding RNA. So let's look a little bit at RNA. What makes it different from our other favorite nucleotide? Of course, it's ribose rather than deoxyribose. That's what the R is for instead of the D. We have uracil instead of thymidine. Probably much more important as far as the function of RNA is concerned, and a lot of these non-coding RNAs, is that you don't always have nice, happy, friendly base pairs. And a nice example is right up here at the top of this particular RNA, GG. GG forms quite a bit in RNAs. So yes, you've got your AU base pairs, but you'll also have some UU base pairs, etc. So many non-standard base pairs that will form and are actually very important for getting you a particular structure. And what does a structure do for you? It gives you function. Exactly. So if we look at, most of the time when people look at RNA, they draw something like this. We've got nice loops, but it's planar. Unless we're in flatland. Anyone know flatland? Yes, a few of you. Good. Most of you not. Everybody should. It's an absolutely fabulous book. Very short, too. But we don't live in 2 dimensions. We live in 3 dimensions. And so these are what the RNAs really look like. Again, of course, is presented in 2 dimensions here, but it's all 3-dimensional structures. So whenever you see something like this, you see these 2 strands, always opposite strand in terms of base pairs with each other. That's actually a helical structure. And so it's a little hard to look at any other way. Again, hopefully not beating the dead horse here. Uracil is basically what? You all remembered your base pairs first time around that I didn't ask you on the midterm? So it's a 5-methyl cytosine that's been deaminated. Right? Yes? No? So it forms 2 base pairs. Again, very similar to thymidine. Thymidine would have a methyl group here and is otherwise normally base pairing with adenine. Why uracil? Turns out the fact that it doesn't have this methyl group here makes it a lot more flexible. So it actually can form a lot more different kinds of base pairs. The other thing, of course, is that now we have a 2' OH in our RNA because it's ribose and not deoxyribose. That 2' OH, as we'll see, is absolutely critical for some of the activities that RNA has, particularly important for splicing, but it turns out for many other activities as well. It's very important that you have this 2' OH. It gives you a lot more possible activities, structures, than you would otherwise have. There are lots of different RNAs that you find inside the cell. In fact, if you look at this textbook, the last version of the textbook, I think it had 4 different kinds of RNAs. Now we're up to about 12 different kinds of RNAs that are being made. The vast majority of RNAs, if you just open up a cell, be it an E. coli cell, be it a human cell, be it a yeast cell, the vast majority are ribosomal RNAs. And this actually made it hard to detect other RNAs for a really long time. Ribosomal RNA is a really nice example of a non-coding RNA because it doesn't code for proteins. We now, of course, know the ribosomes make the proteins. Yeah?
I just wanted to go back to your last slide.
Sure.
Uracil is most similar to thymine.
Yes.
Okay, so just you misspoke and said cytosine.
Nah, but it's the same as a deaminated 5-methylcytosine.
Okay, good.
So that was trying to link back to the last lecture. We're talking about DNA damage, is that that's how you end up with uracil in DNA. You're welcome. Yes, you can also think about it as a dimethylthymidine. But that doesn't happen in terms of DNA damage because that particular bond, the methyl bond, or binding to the methyl group, is much, much more stable than an amine. Great review from last lecture. Thank you. Okay, so the vast majority are your ribosomal RNAs. And you'll notice that this 80 doesn't add up to 3.5 to get 100%. The next most common are, in fact, the transfer RNAs, that again we'll talk a lot more about in terms of translation. Hopefully all of you know about these things. But there's a whole slew of other non-coding RNAs that people have been discovering more and more as time has gone by. And basically we've just been looking at what gets transcribed from genomes. So we'll talk a lot more about the small nuclear RNAs and the small nucleolar RNAs. These are important for modifying other RNAs, microRNAs, siRNAs, piRNAs, LNC RNAs. These are all important in regulation. And so we'll talk about these much more when we talk about regulation later on. Particularly this one down here, the long non-coding RNA, is critical for X chromosome inactivation, one of those long non-coding RNAs. But what we're mostly going to talk about today are the messenger RNAs, which are a relatively small proportion of the RNA that's present in any given cell at any given time. But why we care about them is they're coding for the proteins. And so those coding sequences, the coding RNAs, the exons, these are all going to be making your messenger RNAs. But they're a relatively small proportion of what's there. So to get lots of proteins, and we'll take the example of the histone proteins. Sometimes you need a whole bunch of proteins to do something inside the cell. If you're talking about structural proteins, you need a whole lot of them. So how do you get a whole bunch of some protein and very little of other proteins? You made it at a particular time, or if you're thinking about development, your nose has basically identical DNA in it to your stomach. But your nose and your stomach are hopefully not the same thing. So what makes your nose a nose and what makes your stomach a stomach mostly has to do with what proteins are being made there. And so making those proteins, and making usually lots of those proteins, is what transcription and to some extent translation is all about. Because you can have one gene, say this gene A here, which can be transcribed into many, many copies of RNA. And we talked about this back in lecture 2, where we talked about amplification, or maybe it's lecture 1. Amplification. It's the way that you can get many copies of the genetic information for what's here in this one particular gene. You can also regulate translation, so you get a whole bunch of that. It seems to be a little bit less translational regulation than there is of RNA regulation. And this makes sense if you think about it from a regulatory point of view. You're going to want to regulate this first step rather than regulating later steps, certainly in terms of making larger amounts of things. But what should be really obvious here is that it's going to really depend on where you start in terms of what genes get turned on and what genes get turned on. What makes the RNA, this is the RNA polymerase. A better way of talking about this would actually be DNA-dependent RNA polymerase, because this RNA polymerase, and for this class it will always be the DNA-dependent RNA polymerase, binds to double-stranded DNA and will transcribe that into RNA. This is a cartoon of what that structure looks like. You've got double-stranded DNA. You unwrap a little piece of that, and that gets copied into your RNA. RNA is made just like DNA is made, always from 5' to 3'. So if you talk about being made from 5' to 3', this has to base pair with something which is in the opposite orientation, because otherwise it's not going to base pair properly. So it has to base pair with this bottom strand, what's called the template strand, which is from 3' to 5'. That's the only way these guys can match up with each other. So the actual mechanism is practically identical. You've got your nucleotide triphosphate, gets hydrolyzed, that pyrophosphate gets hydrolyzed, you move to the next step. Big difference with these RNA polymerases is they don't need a primer. And we already talked about this when we talked about the primases. This is why primases are RNA polymerases, not DNA polymerases, because RNA polymerases don't need a primer. They can start basically anywhere. Considerably lower fidelity than DNA polymerases. They don't have the same kind of structure, that hand structure, which sort of closes down on the base pair and checks to make sure that it's got the right geometry. They have a little bit of proofreading, but it's a lot less than you have in terms of your DNA polymerases. So lower fidelity, a little bit of proofreading, but a lot less so. The RNA comes out of the RNA polymerase really rapidly. I always have SS RNA here. A lot of people say, well, RNA is single-stranded. That's one of the things that makes it way different than DNA. Well, most RNA actually is going to fold up on itself and form helices like we had at the bottom of that slide a little while ago. So I like to think of RNA as usually being single molecule as opposed to DNA, which is 2 molecules. Nobody else uses that in the literature, but I find it easier to think about that way. So I don't like SS. We'll say the RNA is being rapidly released. Another thing which is interesting, important, I'm not sure what you want to mention it, but the main thing here is that all RNA polymerases are slow. They don't go anywhere near as fast as the DNA polymerases. And that probably has to do with the fact that they're pulling apart the 2 strands, and you've got these extra processes which are involved. It's really difficult in terms of your geometry, and we'll see some of the actual high-resolution structures in just a second here. Yeah?
And the DNA polymerases were about 5 times that fast?
So depending on which DNA polymerases you're talking about, the eukaryotic DNA polymerases are about 5 times that fast. The bacterial are about 50 times as fast. Yeah?
How is the template strand identified?
How is the template strand identified? The template strand is the one which is going to be complementary, i.e. the one that base pairs with the RNA that's being produced. Now, how is it determined what you use? We'll get to that in just a second. Yeah?
So you're saying that the polymerase is rapidly releasing the mRNA, but that its function is slow? I'm confused because I thought you just said rapid, and then now you're saying that it's...
Okay, so the RNA itself, the RNA doesn't stay base paired with the template strand. It only stays base paired for a very short period of time. Turns out it's the couple of nucleotides. There's a big difference relative to your DNA polymerases. In your DNA polymerases, once you've made that second strand, it stays there. It doesn't fall off.
Okay.
Yeah?
Why is it called DNA dependent?
Why are these polymerases called DNA dependent RNA polymerases? Because they have DNA templates. And next term, when we take virology, all of us, right, there are RNA dependent RNA polymerases. So that's the big difference here. But as far as we're concerned, it's a DNA-dependent RNA polymerase. Whenever I say RNA polymerase in this class, it will be the DNA-dependent RNA polymerase. Next term, I make no bets. So one of the really nice things about this is you can see the polymerase working on DNA here. So this is, in fact, 2 genes right next to each other. These are ribosomal RNA genes, and they're being transcribed. All the little dots down here are RNA polymerases. Because they're relatively slow, you often have lots of them all lined up right behind each other. And so that's what's going on here. These guys have just started. These guys are just about done. And you can literally see these in the electron microscope. So let's talk about, we'll get to your question here in just a second. How do you know what's the template and what's not the template? The probably most important thing in terms of where and how you get your RNAs is where you start. And so in the case of bacteria, you have a particular place that you're going to start. That place is called the promoter. This is also true in eukaryotic systems. That place, again, getting back to Tashney's talk, it's just a DNA sequence. And that particular sequence of DNA, the RNA polymerase needs to get to and put down the first nucleotide and be able to extend it. In bacteria, there's a protein which helps the RNA polymerase get to that specific place. And it's called the sigma factor. We'll hear far too much about factors in the next couple of lectures. Factor just means protein subunit of what you have. So if you think about the RNA polymerase, there are going to be multiple factors that are associated with it. These are proteins that are associated through protein-protein interactions with the enzyme in this particular case. Sigma factor is specific to bacteria. It allows the polymerase to find where to start, which is the promoter. What happens once you have this binding, it's what's called a closed complex that forms. That closed complex, why is it called a closed complex? It's because both of the DNA strands are still bound to each other. Then you have an open complex that forms. What happens then? The strands have come apart. Once you have the strands that come apart, not at all unlike what happens in DNA replication, as soon as the strands come apart, then you start. Then you'll put in the first nucleotides. And so it's that opening of the strands, which seems to be, just like we had in terms of replication, is really important. Now, what's different, replication versus transcription-wise, it's the RNA polymerase that is doing that denaturation step, pulling the two strands apart, whereas in replication it's helicase which is doing that. So the RNA polymerase has this activity. Then once you have an open complex, again, you start initiating transcription. It's just putting in the first couple of nucleotides. What's interesting is this true in bacteria and in eukaryotes is you have this process called abortive initiation. And the reason for that is that unlike the case for all the replication machinery, DNA polymerases are non-processive. They fall off of the DNA all the time. On the other hand, RNA polymerases are highly processive, so they stay on the DNA and will keep going until they get to the very end of the gene which they're making the RNA copy from. Some of these genes, I think the longest gene in eukaryotic systems, I think in mammalian systems, is over 1 million base pairs in length. So basically, this is sort of a bet hedging mechanism whereby you want to be really sure that you want to be making a copy of that whole gene. And so what's called abortive initiation. This happens again in bacteria and eukarya, and it just spits out a couple of nucleotides and then basically thinks, again, to totally over-anthropomorphize, before it decides to move into an elongating stage, and that elongating stage will go all the way through to the end of the gene. Then everything falls apart, the RNA polymerase comes off, the DNA is released, the whole RNA is released, this polymerase goes and looks for another promoter, does the whole thing again. Yes?
So does an amount or like the rate of or how processive polymerase is, does that result in lower fidelity by that polymerase? You're saying the RNA polymerase has lower fidelity?
Yeah, so the question is there a connection between processivity and fidelity? The answer is probably yes, but I don't know all the details of it, but it's certainly a reasonable hand-waving argument. I think it has to do with the exact structure, and you should talk to Dr. Bartlett in the biology department because he knows way more about this than I do. But yes, it's true. It's very different in terms of the processivity and in terms of your fidelity. Now, the other nice thing about the fidelity, if you have low fidelity in your RNA polymerase, it's not something which is being carried forward genetically. And so you can get away with, feels like all of evolution, it's just something you can get away with. As long as it works, it's fine. You can get away with that because those few differences are not going to make something which gets passed along to the next generation. Yeah?
So during the board of initiation, what determines when it stops, and also what determines the go on?
So the question here is what determines what also people call escape or promoter clearance? How do you go from this abortive initiation step to the completely processive form? And the answer is, we'll talk about this a lot more later. It's a regulatory step. And one of the reasons to have all of these different steps here is it allows regulation to take place. And so you can, at any one of these given points, say, okay, we're going to regulate binding. We're going to regulate closed complex formation. We're going to regulate open complex formation. We're going to regulate abortive initiation. Each of these steps gives you an option to regulate the amount of that RNA which you're going to end up getting. And yes, we will talk more about that when we get there. So what do these promoters look like? Again, they're just DNA sequences. So how do you know what that DNA sequence is that your favorite protein, in this case it's going to be sigma factor together with the RNA polymerase, is going to bind to? Well, it turns out that sigma factor and RNA polymerase bind to a whole bunch of different sequences in any given organism, any given genome. As I mentioned before, that interaction of the polymerase with the promoter is going to be a really important part of regulation. And it turns out, and again, as Mark Pashley mentioned last night, is that it's that protein-DNA interaction and how often you have that protein there, which is going to determine how often you're going to go to open complex and making the RNA after that. So it's really all about how strongly or how often that polymerase happens to be at that particular promoter is going to determine how much of that RNA actually gets made. And one of the ways that organisms have evolved to have more of some genes and less of other genes is to have a sequence at their promoter which is going to be better for the polymerase to bind to or worse for the polymerase to bind to. Better binding, more RNA. Less good binding, less RNA. So how do we now know what that is? Well, it turns out you can take lots and lots of sequences and just line them up with each other, this DNA sequences, and then see what nucleotides, what frequency you have particular nucleotides at particular positions. And particularly, these are the nucleotides that you have at strong promoters versus weak promoters. Line up all of these. On average, you're going to have, the actual sequence here is not important, so don't bother memorizing it. TTG ACA, this is true for E. coli, and TAT AAT. This is on average what you get. If you have sequences which are similar to this average sequence, it turns out that's a really good place for the polymerase to bind, so you're going to get a lot of transcription. If you have sequences that are quite different from this, the polymerase is not going to bind there, or bind there a lot less well. You'll have a lot less transcription. There's a couple of notations here that I forgot to mention before. Notice minus 35 and minus 10. And this is getting back to your question about what's the template strand. What are you using? So we know, through various biochemical techniques, exactly where the polymerase puts the first nucleotide in. And that's what's called +1. And since it's always going to be transcribing from 5' to 3', anything that is 3' of that position is going to be plus. Anything 5' of that is defined as minus. And that's in terms of where that nucleotide is. So if you go 10 nucleotides and 35 nucleotides minus, also called upstream of this, that's where you find these sequences that the polymerase is going to bind to. And particularly true for bacteria, they're 10 nucleotides away and about 35 nucleotides away. What is the really interesting thing about 10 nucleotides. What does that mean? One full turn of the helix. So it's going to be on the same side of the DNA as where it's putting down that first nucleotide. Yeah, you had a question?
So then upstream moves towards the 3' end?
Okay, so upstream is going to be 5' of where you start. So if you think about the first nucleotide, and the first nucleotide is going to have a 5' end, it's going to have a phosphate sticking off it, and it's going to extend from 5' to 3', so that's where you're going to be making your new nucleotide. So that's going to be downstream as you're making it, so going from 5 to 3, as far as your new RNA is concerned. The problem is you've got the 2 strands, of course. So if you're now going from the new strand that you've made, your transcript, the RNA, if you're now going in the 5' direction, that's upstream or negative. Okay, so these are the sequences. A lot of people will just draw these. I love these, what they call sequence logos down here at the bottom. How frequently you have one particular nucleotide is how big you have any of these individual letters. So you can see here that, T, A, sometimes, A's, more A's than T's, but not all that frequent here. And then you almost always are going to have a T at this position here. How do you know which ones of the strands to use as your template versus the non-template strand? It's going to determine which RNA is made. Well, if you back up here, one thing you'll notice is that this sequence is only this sequence going from -35 to -10. It's giving you that particular orientation, 35 to 10, because it's only relative to where that first nucleotide is going to be. If you had the same sequence on the opposite strand, that would lead you to transcribe in the opposite direction. These are not symmetric. These are an asymmetrical binding site that you have here. Because of that asymmetry, you're going to end up with either transcribing in this direction because your promoter was over here, or transcribing in this direction because the promoter is over here. And so it's that promoter sequence which is going to determine which strand gets used as your template strand. Yeah?
Do both strands have promoter sequences, or it's not one strand hogs all the good directionality of these sequences?
So, great lead in. Thank you. So the question is, do both strands get used? The answer is yes. Both strands do get used. And very often, what you'll see, and this is just the example from an E. coli chromosome, these sort of, I guess, greenish diamonds down here represent promoter sequences. Very often, you have promoter sequences which people call divergent promoters. So a promoter sequence is going to lead to transcription in one direction, and a promoter sequence is going to lead to transcription in another direction. And of course, it's going to be using now different strands as your template strand. And one of the nice things about this, again, gets back to regulation. Because if you've got the promoters right next to each other, something you do to one is probably also going to be influencing the other one as well. So it makes sense to have these guys together. But it varies. You get both strands that get used. So what does the polymerase actually look like? This is from the 4th edition. I was going to pull up a newer version now. We now know the structures of particularly the RNA polymerases in the initiation state to very, very high resolutions. So basically know exactly where each and every atom is in the structure of all of these polymerases. This one is a slightly lower resolution structure here. But a couple of take-home messages from here. One is it does not look like a hand. It doesn't look like all of those DNA polymerases that we've looked at before. You can see that there's a very short piece of RNA which is interacting with the DNA, coming out of the RNA polymerase complex. And so this is getting back to the question about how rapidly you're getting the release. It's only a very short piece of RNA that is in the active site and base paired with the DNA at any given time. So these guys unwrap the DNA, untwist it from each other, put down the nucleotides, it wraps back up very rapidly. The way that it does that is that there's what's called the rudder structure in the RNA polymerase, which basically sticks down between the two strands, and as the polymerase moves along, that just basically peels apart those two strands relative to each other. So it's not like the helicase that has a circle around it. It's that rudder where it gets stuck in, and that gets stuck in just when you have open complex formation. Remember, so that first step that you have in transcription is pulling those two strands apart. Those two strands come apart, the rudder gets stuck in between them. Yes?
So I assume there's still the issue of needing topoisomerase farther down the strand as you pull it apart?
Yep, we'll get to that. So you do need topoisomerases in transcription as well. The advantage that you have with transcription is you're pulling them apart, but you're putting them back together again very rapidly. And so it seems to be less of an issue than you have in replication. But you do need topoisomerase, and we'll get to that in just a second. Yes?
What about ATP? Helicase is pretty ATP dependent, but this isn't.
Yeah, so when the question here is, what about ATP hydrolysis? And certainly helicase requires huge amounts of ATP hydrolysis to pull those two strands apart. The RNA polymerase itself doesn't need extra ATP hydrolysis above and beyond the adenine triphosphate that it's using to extend with. So there's no extra ATP hydrolysis at this step. We'll get back to that a little bit later on. But it's not like when you case the helicase, we have to have huge amounts of ATP hydrolysis the whole time as you're doing it. Okay, so this is what happens as the polymerase is moving along here. And in fact, just like the case with the DNA polymerases, it's probably not the polymerase moving along the DNA. It's probably the DNA moving through the polymerase. But it's all just a matter of your point of view as far as that's concerned. So what happens when you get to the end of where you want your RNA polymerase to be making things? Now you have to terminate. So we've got initiation, elongation, and termination. Termination happens in a number of different ways. Probably the best understood of these is something called row-independent termination, which means you don't need an extra protein. It's just a sequence-specific interaction. You have a whole stretch of U's, which get formed in your RNA. U's are normally base pairing with what? A's. How many hydrogen bonds do you have? 2, relatively small numbers of hydrogen bonds. So this interaction here is a relatively weak one. And you remember in the structure of the elongating polymerase, it's probably not even as many here as they got 7, 8 nucleotides base paired here. It's probably more like 2 or 3 that are actually base paired. And so that's a relatively unstable structure. And what it turns out in this particular kind of termination is there's another structure that the RNA loves to form right next to that. And that's called a helical structure, and there's the base pairs are now self-complementary to each other. So what seems to happen is as the RNA is being made, and remember this RNA gets outside the polymerase really quickly, it's going to form this hairpin loop structure. That's what this is. Remember, it's a helical structure with a loop at the end. And that basically pulls the RNA out of the polymerase. And so some people will say this is a speed bump in oil. The speed bump is this structure, and oil is this greasy stuff right here. So it hits something, it's a little oily, and so the car flies off the road, the RNA falls off of the polymerase, and you restart the whole thing. Yes?
Is there some method in which it gets prevented from forming initially before termination?
So these hairpin loop structures, the hairpin loop structure here, the question is basically, is there a way that this is prevented from forming before termination? And the answer to that seems to be, I'm not sure why this is dying on me all the time, is that this is now sequence dependent. So it has to base pair here. And most of the time in your RNA, you're not going to have something that's going to form that nice, stable base pairing interaction. So it's a very sequence-specific kind of process that's happening here. This is very specific to bacteria. Turns out it doesn't happen in eukaryotes. We'll talk a little bit more about eukaryotic termination later on. It has to do with splicing, which doesn't happen very often in bacteria. This is bacterial transcription. The big differences here are going to be your sigma factor for your specificity and the termination with these specific sequences. Questions about the bacterial transcription? The mechanism is practically identical in eukaryotes. Oh, yeah. Yes, so the polymerase falls off as well. If there's no longer the RNA associated there, the polymerase is going to fall off as well. So it seems that the release of the RNA is really the most critical aspect about holding that whole thing together. So in eukaryotes, again, these darn eukaryotes, they're slow and big and all these other kinds of things. Much more complicated and confusing, but these guys have 3 RNA polymerases. 1 wasn't enough for them. And these are, again, DNA-dependent RNA polymerases. RNA polymerase 1, 2, and 3, again, incredibly creative naming process here. RNA polymerase 1 makes most of the ribosomal RNA genes. And you remember, those are the RNAs that are the most prevalent inside any cell. RNA polymerase 3 makes most of the tRNA genes and a number of small RNA genes, but the granddaddy of them all is RNA polymerase 2. And we'll spend most of the rest of the class talking about it. All of these RNA polymerases we'll see in just a second are very similar to each other in terms of structure, which means they're going to be very similar to each other in terms of function, because they all do basically exactly the same thing. These polymerases in eukaryotes are all very large numbers of subunits, 12 to 14. They all vary slightly relative to each other. In bacteria, there are only 5. However, even though there's only 5 subunits in the RNA polymerase in bacteria, it turns out that these subunits are homologous to each other, which means they have a common ancestor, and they're still homologous enough that you can see sequence similarities. So all of the largest subunits that you have in eukaryotes are also similar to the largest subunits that you have in bacteria. Same thing is true here for these other subunits. One thing that you'll notice that is missing here for the bacteria one, there's no sigma. So that's the difference that we have here. Probably not get to this today, hopefully next week. Turns out that the archaeal RNA polymerase is much more similar to the eukaryotic RNA polymerase II than the bacterial and archaeal polymerases, which is one of the reasons that studying archaea is really cool. But they've only got 1 polymerase instead of 3. Not surprisingly, because these sequences are so similar to each other, the structures are also very similar to each other. This is the eukaryotic RNA polymerase II structure. Here's the archaeal RNA polymerase, and here's the bacterial RNA polymerase. So if you squint a little bit, they're basically identical to each other. These guys have got a little bit of extensions here, and these guys clearly have some extensions up there. So let's focus on RNA polymerase II. This is that structure here. Again, it doesn't have a sigma factor. Sigma factor is important for getting the bacterial polymerase to interact with the promoter. Well, you need promoters. You need a place to start in eukaryotes as well. How do you get that to happen? You have general transcription factors, and we'll talk about all of these in just a second. They're called, again, factors, just means they're a protein. Transcription, because they're involved in transcription. General, because you need them at multiple promoters. We'll talk about specific transcription factors a lot later on. And again, incredibly creative naming, called TF for transcription factor, TF2, because these particular transcription factors are working with RNA polymerase II. So if you had a TF3, that would be interacting with RNA polymerase III. A TF1 would be interacting with RNA polymerase I. A, B, D, E, F, and H. And we'll see what these guys do in just a second. RNA polymerase II, just like all of the other RNA polymerases in eukaryotes, also clearly have to deal with nucleosome-bound templates. They're dealing with chromatin all the time. What do these GTFs do? Again, they get the polymerase to the promoter. How do they get the polymerase to the promoter? The promoters, just like you have in bacteria, they've got DNA sequences. Those DNA sequences are going to interact with some portion of the polymerase and allow the polymerase, and actually through protein-protein interactions, to get the polymerase to bind to the promoter. The main important aspect of this promoter, again in the DNA sequence, is what's called the TATA box. Why TATA box? Because it's TATA, actually highly rich in TAs, and box just because it's a particular place in the sequence. People talk about that as being the double strands together. So the TATA box, again, incredibly creatively interacts with the TATA box binding protein. Wow, TBP. TBP is part of this TF2D complex. So it's a whole bunch of proteins with TBP in it that bind to the TATA box. Once they've bound, then you have interactions in some promoters with TF2A, but in all promoters with TF2B. TF2B binds to the TF2B recognition element, so it's another sequence which is there. And then once you have TBP, TF2B, then you have interactions with TF2H. These were all named because they were just different fractions that came off of a column, but it's easy to remember what TF2H does because this H stands for helicase. Actually, it's purely coincidental that these two things happen, but it has to have helicase activity. So this is a little different from the eukaryotic, so the bacterial polymerase, because in the bacterial polymerase, what had the helicase activity? The RNA polymerase itself. So here, you may or may not remember in the structure, the RNA polymerase, these little ends that hung out a little bit further than in the eukaryotic polymerases. It turns out that those have that helicase activity. So helicase activity and also kinase activity. So if it's got kinase activity, what does that mean? It phosphorylates something. What does it phosphorylate? It phosphorylates the RNA polymerase. And in particular, what it does, it phosphorylates a specific part of the polymerase. And this is a part of the polymerase which is specific to RNA polymerase II. Phosphorylates this what's called C-terminal domain of the largest subunit of RNA polymerase II. What do phosphates? Why would you phosphorylate something that's associated with DNA? We already talked about this before. So phosphates, what is the DNA made of? I didn't bring my model today. Phosphates on the outside. What do phosphates do when they get close to each other? Repel each other. You asked about abortive initiation a little bit earlier on. This is one of the ways that you deal with abortive initiation. You phosphorylate the RNA polymerase, so now it's going to be pushed away from the DNA, and it seems that abortive initiation, that phosphorylation that happens, leads to escape from your abortive initiation process. There are other factors in here, TF2E, TF2F, etc. You will notice that I don't have them listed here. I would not try and memorize what they're doing, even though a very good friend of mine did her PhD on trying to figure out what the heck this protein was. So just another way of looking at this, TF2D, we have talked about this quite a lot, contains the TATA binding protein, but also a number of, again, ridiculously creatively named TATA binding protein associated factors. Great. Okay, so they're just more proteins that are associated with TBP and are part of the TF2D complex. We'll see what they do later on. TF2B interacts with a specific promoter sequence, which we'll look at on the next slide, and it helps put the polymerase in the right place. TF2F and TF2B we haven't talked about, so don't worry about them. TF2H has helicase activity, so it's unwinding the DNA, and has phosphorylation activity, it's kinase activity. So anyone who's got their notes here can just make a big X through these two guys. They're absolutely critical in terms of how they work, but we're not going to talk about it in more detail here. So if you look at the promoter sequences for eukaryotic promoters, most of them are going to have this TATA box, and all that this is TATA. TATA is great, and it binds to the TATA binding protein, but TATA has a problem. And that is, if you look at TATA on this strand, what's the sequence on the opposite strand? TATA. How do you know which strand you're going to use? You don't. So you have to have something else above and beyond this TATA box, the TATA element. And usually that's this TF2B recognition element, When people talk about elements, it's going to be a DNA sequence. We talk about factors, they're going to be proteins. Again, I did not make up this terminology. Mea non culpa here. So B recognition element, again, it's a DNA sequence which the TF2B protein interacts with. We also have a downstream promoter element. Getting back to your question, upstream versus downstream. This is now going to be plus relative to where you have your transcriptional start. So it's a downstream promoter element. And then the initiator element. These guys, the TATA binding protein interacts with TBP. But wait a minute. TBP is part of TF2D, and it's supposed to be interacting with the downstream promoter element and the initiator element, but that's 30 base pairs upstream here. What the heck's going on here? Well, this gets back to how Watson and Crick were not completely right. They had this wonderful B-form linear DNA structure, but one of the great things about DNA is it's incredibly flexible. So one of the things that was found when people looked at how this TATA binding protein was associating with DNA is they found that the TATA binding protein made this big bend in DNA, which is kind of interesting, and I'll bring the cover of Nature where they originally published this structure of the TATA binding protein. It actually had normal double-stranded DNA going right down the middle, totally wrong, and then about a month later they came out with this structure, which is the correct one, where you have the DNA very severely bent here, and basically what that does, it takes the downstream sequences and brings them right next to the upstream sequences. So the other proteins in the TF2D complex that come together with TBP, those are going to be able to interact with upstream and downstream sequences simultaneously. So that's your first step. Now remember, you had TBP, TF2D binding, then TF2B, then the polymerase, then TF2H. So all of these processes coming together to assemble your RNA polymerase at the right place, at the promoter, so that you can get open complex formation, abortive initiation, etc. This may seem complicated. It's actually a vast oversimplification of what's going on at most promoters. So you may notice that there's a whole bunch of other gray blobs in here, plus this big purple blob here called the mediator. And then we'll talk much more about these activator proteins later on. The whole complex here, starting transcription at a eukaryotic promoter, is more than 100 different proteins. They're involved, getting all of them to one promoter and doing it in the right way. Now, this would be hard in and of itself, but if you think about the eukaryotic DNA, it's got all of these nucleosomes that are associated with it as well. So it's not just all this stuff that you have to have here to get your polymerase together, but also you have to deal with all of the nucleosomes. So there's yet another set of the complex which is there. Transcription elongation is just getting back to one of your questions over here about the topoisomerases, is that you do clearly need to have topoisomerases in front of and actually behind your RNA polymerase. The rest of this is not critical. It's topo2 or topo1. The big thing to mention here is that positive supercoiling, which is overwrapping the DNA, makes it harder to pull the base pairs apart. And that's what the RNA polymerase has to do in order to be able to transcribe. And so overwinding is bad. You need to take care of that. And that, again, can be either topo1 or topo2. DNA gyrase does the negative supercoiling, which is unwinding the strands, which helps pull those two strands apart in terms of getting transcription to take place. We'll talk about archaea on Monday. Yeah, it's Friday. Weekend. Yay. We'll see you on Monday.